this morning. You've already actually had, I believe, a nanophotonics seminar earlier this week, and I think you've also seen some presentations on sort of long wavelength biophotonics. So some of the stuff you're going to see today would be sort of maybe a little bit more in depth on some topics that you've already been introduced. Our first speaker today is Scott Howard from the University of and Scott's going to be talking about nonlinear optical microscopy. Scott actually has a background also in the mid-infrared like me, but that's a PhD, which he received at Princeton University. He then went on to do a postdoc at Cornell, in Professor Schu's group, right? And he's been at Notre Dame now for 3 years.
Great, sure. Yeah, so I'm sorry, but like Dan was saying, my main work is basically in two different sides. I do mid-infrared, some type of biosensing, some chip stuff, laser side stuff, and then on the near-infrared we use nonlinear optical microscopy. And next week you have my postdoc advisor over here. He'll be talking about some advanced topics of that. I've tried to do a little bit of both when I do my research group. In the mid-infrared, you get all these vibrational signatures, and you can get very good quantitative measurements. And in the near-infrared, when you use nonlinear microscopy, you can get some very nice measurements of very high-resolution, three-dimensional images of tissue. And so I've been taking approaches thinking about ways of getting quantitative measurements of chemical species, at this hundreds of nanometer scale you can get with nonlinear microscopy, and so we'll have a little introduction to that. And then since this theme is nanophotonics, you do a lot of like plasmonics and nanoparticles. It took us a chance, for the past week to kind of catch up and read a lot about how people are actually using nonlinear optical microscopy with these nanophotonic probes to do this nonlinear, nanophotonic plasmonic microscopy hybrid, and there's actually some pretty impressive stuff going on here. So I hope to give you a good introduction to the fields that I work with with this nonlinear optical microscopy for basically nano imaging, nano detection, and then also this plasmonic stuff that's going on. Okay, so the talk basically you have three parts. We're going to talk. So is there a laser pointer on this? Yeah, there's a green laser pointer. Okay, we're going to talk about just basically this motivation, like where do we need to go? What's currently standard technologies that's out there, and what's some things that would be missing or something we can contribute to. And I'll go over some of the fundamental physics, understand what can we do, how can we get there, where are the little knobs we can turn and play with. And then finally do the engineering. What's a bunch of different experiments and setups we have from literature, from my group, and from some stuff I've done in the past, and kind of showing some things that people are doing that can get us the information we're looking for. And since you guys are in a biophotonics summer school, you've been probably getting this, drilled into you for the past couple of days. But if you look at biophotonics or just biology in general, we have these different length scales, and we need information on these different length scales. And at every length scale, we get something interesting about biology, and you also can use physics in some interesting way, starting with the whole body, which is in the meters. The technologies you can use, MRI or X-ray can pretty much be used for either. Then you want to go down to the cellular or subcellular level. We start getting into this area where we can use optical microscopy. But the interesting thing about optical microscopy is we get this physics information back about electronic or vibrational modes, but we start running into this limit of our resolution. I'll talk a little bit about that in a bit. And so if you really want to get inside of the cell or get information on metabolism in a very local environment in a tumor or in vasculature, you really got to start going to the hundreds of nanometers. And if you're really interested in molecular, how proteins can signal each other, you're going to be in the ones to tens of nanometers, and you have to be in the scale of the proteins. And this is where we start moving into nanophotonics. And I'll mostly be talking about nanophotonics in the area of hundreds of nanometers. If I think later today, I think we'll be talking more people who are doing the science that can really go into the smaller scales. And like I said, we're doing this imaging. If you want to do something in vivo inside of a living animal, we have all these balances. We're trying to trade off resolution, contrast, specificity. And it's a typical thing to say, okay, optical microscopy, we can see all these great electronic modes, gives us some specificity based on this spectroscopy that we get back. But we're limited by the wavelength, so we have this trade-off, and then how deep we can image is limited by our scattering, over an absorption over tissue. And so we'll address some of these. And there's little plugs for, Dan will be talking a little bit about vibrational spectroscopy today and how we can use vibrational states to image biological molecules. And the next week, we'll talk about penetration depth and scattering, as Professor Schu I'll be talking a lot about how can we go all the way through the cortex of a brain, or go even subcortex using optical microscopy techniques. And I guess if you're going to start talking about optical microscopy and thinking about, well, what are we currently doing right now? We could just take a brief look at the gold standard of microscopy, basically of tissue diagnostics, would be the H&E biopsy. And this is the slides you would get. Basically, it's a tissue that's stained, and you can have a highly trained medical doctor who can look at this and say, with extremely high accuracy, okay, I see a tumor here. It's well localized. It's not really spread very much, and I can diagnose the grade and the level of inflammation. What type of cancer is it? Is this metastatic from kidney, and it's actually in the brain? Or they can look at all these different things to come up with this diagnosis and a plan for treatment. Okay, but to get these good samples, the problems that us as engineers are trying to solve are, well, you need to sometimes take multiple biopsies. You got to go in there and actually cut out the tissue. We have to fix or preserve it, so we have to put it through a chemical process, driving out all the water, replacing, cross-linking the proteins, basically making it preserved so it won't rot on you. And then we have to stain it. We introduce external contrast mechanisms so that we can see where's the interesting features in the cells or in the tissue. And then we can then have to mechanically section it. So we have this, this mass, and we basically put it into a giant deli slicer, well, a small deli slicer that just slices it up into a couple micron slices, and we put them on slides. And then we have a team of several, pathologists who sit around with these slides and look at them and do your diagnosis, and and can aid, the other doctors who are planning the treatments. And typically, the external contrast agents we use would be, this hemotoxylin, which is going to the nucleus, making it blue. It also goes for, red blood cells turn very red from it, and eosin, which turns the cytoplasm and the extracellular matrix very red. Sorry. Okay, so the things that I'm going to talk about and trying to address a little bit are these areas of, well, what can we do to reduce the number of reduce the need to cut out things. We want to look at stuff that's inside of tissue without actually having to remove the tissue, so we can look at it while it's still alive. We want to not fix it, and we want to, I will use some stuff with some external contrast mechanisms, but we also want to look at, what your tissue can do just naturally without having to label it, because there's a lot of information in there. And yeah, we don't want to have to slice up your tissue. You want to keep it intact while we're doing this, which is great also not just for diagnostics, but if you're doing mouse model studies of different diseases, you want to be able to see this progress over time and not have to chop up the mouse every time you need some image. And so this is kind of a map of kind of the standard technologies that are out there for medical imaging. And again, it's a trade-off between penetration depth and resolution based on what techniques you're using to look at your tissue. And MRI and CT X-rays are up here for your entire body, but you don't have very good resolution. And then you have this kind of this frontier where all your technologies lie across. And if you're looking at technologies which are as good as H&E or can compete with H&E type of histology biopsies, you want to go at least the same optical resolution of a microscope, and you want to have at least as deep as the epithelial cells. So you want to be able to go some penetration depth through tissue to get something more information than just a pretty picture. And so if you kind of draw these lines on here, you see that there's a little quadrant here where there's some interesting technologies where we can beat H&E resolution, and we can also go deeper than epithelial cells. And one of them is there's multiphoton, and this is kind of motivating. We'll talk a little bit about what this multiphoton is, and then how it's being extended for chemical sensing, specific chemical sensing. Okay, so from this map, kind of recapping what we're missing. So we want to do in vivo imaging. You don't want to have to do H&E slide and chop up our sample every time. We want 3D, so natively inside of the samples. We want resolution superior to H&E staining, specifically if you can get down to the tens of hundreds of nanometers. And also we want something quantitative. You want to be able to say, okay, there's this concentration of hydrogen sulfide in the plasma at this location with such partial pressure. And so what we're going to try to do then is do clever physics to try to beat this this plane wave focusing diffraction limit. This is nanophotonics and nonlinear optics specifically. And we're going to talk about quantitative nanoprobes, which are useful for measuring this specific chemical species at this nanoscale. Okay, so now we're going to talk a little bit about the fundamental physics. I'll talk, just a very quick, 2 slides on, the diffraction limit to get it fresh in everyone's mind again. We'll talk a little bit about nonlinear optics and a little bit about plasmonics, plasmonic structures on nanoscale, and kind of give the structure for what the technologies that we'll base our diagnostics and imaging and platforms on. Okay, so diffraction limit. From a physical optics class, you can start off with a Fresnel-Kirchhoff formula. Basically, what it shows is that if you know the scalar field along some surface, you can calculate what the scalar field is at some point inside the surface. And okay, you can do some quick approximations. And if you put your source, kind of even with your aperture and really far away, it comes pretty much collimated by the time it hits your aperture, and it simplifies down to this expression here. Basically, it says that at any point p, I can find what my vector is here. I know what this angle is here. I just calculate this integral, and I'll find what the field is. Okay. So if I put p, this p right here, kind of far from S, and also make this angle kind of smallish, you can reduce this all down to be proportional to this one integral. Okay, if you then this integral here is just the integral over your aperture. This S here is the surface of your aperture, and e to the IKS is just this phase that accumulates between here and here. So if the amount of phase that accumulates, we can represent this S, which is this distance between your aperture and your or a point in your aperture and your point you're interested in. You can just represent it in terms of angles and distances, and so now we can find what our scalar field is as a function of angle, and we can then say, okay, well, let's use a lens which maps angle to position, and we just plug that in there. You can get a simple integral here, which ends up being the solution to this is basically a Bessel function. It's basically it includes something with this airy disk pattern that you get all the time. And so this solution, get the intensity profile here, where your parameter for your Bessel function is going to be some function of q, which is what I'm putting as this distance displacement here, and then parameters of your system. What's your focal length of your lens? Will be capital R. D will be your diameter of your aperture, wavelength, and pi constant. Okay, so then you get your disk, and so we can plot. This is normalized to this x function. This is a normalized intensity, and you can numerically find that the minimum occurs at this resolution limit here, which is a very common resolution limit. You can express this in numerical apertures or whatever. So this is just basically this fundamental limit that if you have some aperture and some lens, you will get a focused spot that will have this form, and you are stuck with that size from this very simple plane wave expansion. And so this is kind of what we're kind of limited with. And so if you want to really learn about these electron transitions, you want to focus light down that will excite electronic transitions that are unique to each molecule. And we want to do it on the nanoscale. And now we're limited by our wavelength. On this expression here, we basically, if you're on the order of the wavelength always. How do you get down there? And so there's different technologies. This is just a 3 I just put up on this quick slide. I'll talk about plasmonics. Near field, you can have some scanning probes. I won't really talk about that here, but it's another way you can do this. And then there's also some ways of using nonlinear optics. And so I'll mostly talk about plasmonics and nonlinear optics here and introduce what we can do with them. Okay, so I know you've had a lecture already on plasmonics, and we have 2 more after me of people who are really into plasmonics. This is something I know about and is learning about for this week. But the general idea here is that if you have a TM wave on a dielectric metal or air metal surface, you have this coupling of this wave with the carriers in the metal, and you end up with a very localized, evanescently decaying or exponentially decaying wave along the surface of the dielectric and into the metal. And the interesting thing about this is that this localized field, this is this delta D here. This delta D is on the order of like hundreds of nanometers. This is very nice. So if you put in light at 1.5 micron, put in light at 500 nanometer, you end up with a localized field in the orders of hundreds of nanometers. Okay, so you can really have an intense localized field right there, and you can do sensing of proteins binding on cell membranes or something on the order of hundreds of nanometers. It gives you this enhanced resolution. But it has this additional kind of neat feature here that if we can get some nanoparticle or some metal sphere in this case, where the size of this metal sphere is much smaller than the mean free scattering length of our electrons in this metal, and we use a wavelength that's much larger than the metal sphere, basically you can have a sloshing of these electrons that will never scatter. You know, this like almost like a cavity, pretty much, that you have this resonant oscillation as electrons kind of slosh back and forth. And this oscillations then, because you have this resonance, will have a very specific wavelength of energy of light that will correspond to this resonance. You get absorption. You get this sub-wavelength confinement of very intense electric fields on either side of the particle, depending on what phase of the light is currently an incident on the particle. And then so you have this very tight confinement and very high field enhancement. And there's a lot of science going on right now, you know, using these gold nanoparticles to try to do really small measurements of things right on the surface here, which is going to be on the orders of nanometers, single nanometers. Oh.
Yes. So this phase distribution of the nanoparticle, like the length itself, what does it correspond to?
Oh, sure. So you want the phase distribution, so across the nanoparticle, or?
Yes. The red ones are like so much.
Yeah, yeah. So basically, if you almost can picture this as, in this case here, you know, you're hitting this resonance. It's almost like a static field at this point if it's so much longer than the wavelength. And this thing's so small. The reason why it's so much larger now, it looks like a static field. So we're just biasing it, and we're just pulling all the electrons to one side and the other side. So across here, it's, you know, it's basically it's as much separation as you can get for the field that's present at that time. So when you're at the null, when you're at the 0 point electric field, you get no distribution. At the peak of the electric field, they separate out more. And yeah, so I think probably the next people are more experts on this. But is that kind of what you're asking? Like, you know, how much distribution there is across the nanoparticle?
I from the picture, I kind of see a wave, right? Maybe the static wave, whatever. So the length is still like of this wave.
Sure. So so this wave here, you know, I kind of have this picture of this wave going. It's you know, if this is 50 nanometers. We're sitting in a plane wave that's going to be on the, you know, it could be, you know, hundreds of microns, millimeters. So you're you know, 3 or 4 magnitude bigger than your particle, and that's what I'm saying. It looks like a static wave. Yeah. Okay. Oh, and also, yeah, questions are, you know, you can please ask questions. You know, I'm very happy to just talk about stuff. Yeah, professors, that's our job. You know, we're professional talkers, so it's like, that's fine. Okay, and so now if you have these nanoparticles, we can start playing with them, and there's a lot of research going on here when you can get different nanoparticles, and you can change different shapes of them. So you have different nanorods of different aspect ratios, you have different nanospheres. All these are of gold, and yet they all can absorb light at very specific frequencies. And these frequencies are a function of how big these particles are, and then how much what wavelengths are required to have the electrons slosh properly back and forth in this resonant condition and get absorption. And I think you're all getting my slides, and I'm drawing a lot from literature review papers. I probably have maybe 30 or 40 references in this talk, so please go check them out. This was a good paper. Why gold nanoparticles are more precious than pretty gold is the title of the paper. And I bet they wanted to name this paper "Why Gold Nanoparticles Are Worth More Than Their Weight in Gold," but then it's kind of this recursive statement that doesn't go anywhere. So they had to change it probably into something that makes sense. Pretty gold. Okay, yeah. So so you can play around with these different shapes, and you can tune them to be resonant. And it's good to look right here. In this example we have here, they're tens of nanometers shapes, sizes, and they're going between 500 nanometers all the way up to 1200 nanometers. You're in the reddish to the near infrared wavelength range, and that comes in very important later. These are just the natural absorption resonances for this, and it's a function of dielectric constants and the plasma frequencies and the sizes of your stuff. Okay, so that gives you a little bit about plasmonics, and we'll use this later. And the other thing is saying, okay, well, are we really diffraction limited based on what we get from a plane wave incident on a material? And if I drew this airy disk here, I have this dashed line. Okay, that's what it looks like. But imagine for incident on a material where your response that you get back from it is not a function of intensity, like you would get from just a pool of fluorescent dye. You can put a flashlight in it. But it's proportional to intensity squared. So you can picture the airy disk here, these higher peaks, you kind of pull it up. And then these lower peaks don't really get pulled up as much. And you actually end up something that's pretty Gaussian. This little red line here is the actual function, and this dashed line is the Gaussian fit to it. And you can see that there's a narrowing of this width to the point where you can actually see something on the orders of hundreds of nanometers optically. if you can make something that's nonlinear. And so I don't know how much background we have in nonlinear optics, so I'll give a 4-slide crash course in nonlinear optics, so you can kind of get this appreciation of where does this come from, like how can we get this. And we can start off with actually pretty semi-classical, purely classical point of view and saying, okay, well, we have some electrons, or we can put some electric field across them, and we can separate them out like a dipole. So we can have, if you put a positive field here and negative one there, electrons will move over towards it, and you end up with a positive charge on this side, a negative charge on this side, and your neutral molecule is now a dipole. It's basically like a mini antenna in this. So we want to study, well, how can this electron, these dipoles be formed, and what is the the frequency response of these dipoles, and that can kind of get us information on what light can be radiated by these dipoles. Okay, and so you can say, okay, let's look at the polarization. It's basically going to be, well, what's the average distance in some volume that these things can separate by? What's the charge on each of these dipoles? And what's the density of these dipoles? And from here, we can get some overall average polarization of your material. And then we can say that, okay, well, we know if you apply electric field, there will be a response. And so let's generally just describe it as a Taylor expansion of electric field. Okay, and then we'll have these coefficients, which will be the first order, second order, third order coefficients to this Taylor expansion. And we'll see what happens when we have this type of structure. Okay, so let's put in some monochromatic light. It's just cosine omega t. Omega is your angular frequency of your light. And plug it in here, and you get the cosine term, cosine squared term, cosine cubed term. And then we can have fun with trig identities. And you just move some of the terms around, and you get a few more terms, each of them at different frequencies. You get a DC component. This is called optical rectification. And actually, you can do this in semiconductors. You can make this, and you can actually measure this voltage. You put light on something, you can actually measure a static voltage, even though you have an AC field. We have a linear response. This is what we're currently used to in everyday life. It's just, this is the same response that you would get from, Snell's law. You get light refracting through that window over there would give you this linear response. And then we had some, I'm just calling this a third order response for now. We'll discuss this a little bit more. Some chi3 term in it, but it's also at omega. It's at your fundamental frequency. And then we had second harmonic generation. We have something that's going to be generated at 2 omega, double your frequency, and then third harmonic generation, something at 3 omega. And then the higher order terms as well. But we'll just stick to 3 for now to talk about this. Okay, so I'm putting in light at omega. And here I'm getting a linear response at omega. And I get something else at omega also, with some electric field cubed in front of it. Okay, so let's look at just this component of the polarization that is at omega. And you end up with this expression here, which is the 2 terms from the previous slide. And I just write this as like some linear susceptibility and some nonlinear susceptibility, just to find new terms. And we say, okay, well, what's the index of refraction when you have this case? And you can say, well, the index of refraction is square root 1 plus susceptibility, and I put these in here. We do some square root identities, and we end up with a function of index of refraction, which is proportional to 1 plus or chi3, or this nonlinear susceptibility, and electric field squared. So basically, we have an index of refraction, which is a function of our original index of refraction, of whatever material you're in, plus something which is a function of intensity. And this is very important. This is Kerr effect. You can do it for many different optoelectronic devices are employing this. And really interesting physics. But if you think about this is a chi3 here, and if it's perfectly real, then this is going to be perfectly real, and then you just have just basically a change of index of refraction. But if you can have a complex chi3, you can think of a complex chi3 as basically some types of relaxation or absorption inside of your system. You would get a complex N2, which then will lead you to an absorption coefficient. So this is just physical optics, basic optics. Complex index of refraction means you get absorption. So right now, you just have a system where the absorption coefficient, how much energy is absorbed by my molecule, is a function of intensity. So it's a nonlinear system. We have to have intensity there first. And the brighter that intensity is, the more it will get absorbed. So we actually have created our nonlinear system, which would cause this resolution enhancement. And all these chi3s, you can, there's just an old dipole matrix elements in quantum mechanics. You can, if you're taking a nonlinear optics course, you can actually go through and calculate for a system what these chi3s are. And they're real physical, you really have a physical response here. So this is a real effect. Once we started making lasers where intensity can get bright enough that the small N2 starts dominating N, we can actually start seeing these effects. Okay, and we'll take it into the quantum a little bit and think of, okay, what's going on in this quantum scale? If you have some ground state and some excited state, when you just do a normal linear excitation, a photon comes in, excites from the ground state to this excited state. There's probably some relaxation between the two, and then it emits a photon down at some energy that's a little bit lower than what was put in. With two photon, essentially you said, we have to have two intensity squared. You have this intensity coming in, so you need two photons that will simultaneously be absorbed. Same relaxation, the photon comes out. It's important to note here that here the photon that comes out is actually at a shorter wavelength, higher energy than the photons that went in. And these processes occur both with dyes, and also a lot of tissue just does that on its own. So this type of signal is kind of common on its own. And I'll talk a little bit about that later. And so here are some slides. I have them here so you guys can download them and read through them. This is a very, very quick description of how you get axial confinement, how we can see in 3D with nonlinear optics. So I'll just go quickly through this, and there's some equations in here that you can read on your own if you're interested in that later. But basically, I just showed that typically your fluorescence, if you think about yourself as your dye or solution as someone playing pool, you have these dye molecules all over the place, and you're shooting a pool ball. The probability of hitting a pool ball is simply going to be proportional to the density of cue balls you're hitting times the density of these other balls you have on the table. Again, I'll just tell you the number of collisions that will occur is just linear proportionally. But if you want to do a two-photon event, you need to find, well, what's the, you're hitting a bunch of cue balls at the same time. You want to see what's the probability that two cue balls will hit one of your other balls at the same time. Well, that'll be proportional to, well, what's my density or a probability of there being a dye present, plus what's the probability of there being another cue ball present at the same time, times what's the concentration of flux of my ones that I'm hitting, and you can just do some you can just do some proportionalities, and you can find what this expression is, and then you can expand it to n photon process, and what you end up with is, well, the number of events that occur, the number of these two photon absorption events and then thus emission events will be proportional to, so this is the power to the n number of photons. If you have a two photon process, it would be power squared divided by the area to the n number of photon process minus 1. So if it's a two photon process, it would be power squared divided by area times concentration. And then a three photon process will be power cubed divided by area squared. And then a linear process will be power divided by, it's just power. There's no area dependence. Okay, and so this is our expression, kind of showing how much our fluorescence response is a function of position. And so we basically need to know, well, what's the power as a function of position, or area as a function of position, and concentration as a function of position. And so if you have a one photon response, our power is going to kind of exponentially decay. Our area does some function like this. So the one photon response, our fluorescence as a function of position is simply going to be the power that's at that depth. And you end up with kind of this blur. So if you put in some excitation, you actually get emission all throughout your sample. You don't really get much information back in the way of where this was localized in the axial dimension. But if you do the two photon, you have a power squared divided by area. So I have power divided by area. I end up with a very localized spot. And this localized spot will give me this little point right there. And I can now know that all the light that comes back that I receive had to have come from this little tiny speck, which is so small you can't even see it on the screen apparently. But there's a spot there, and that's what you're looking at. And then here's actually an experimental demonstration. I guess got white light coming in here, and you can see this little bow tie shape you would get from fluorescence. I put in some infrared light, so I can get a two photon absorption in some fluorescein dye, and I get a localized speck in 3D. This is actually super saturated. The actual speck size is less than a micron in diameter. And so this is something we can raster scan inside of tissue and get a three-dimensional reconstruction of what we're looking at inside of tissue. And to show that we actually get improved resolution, here's the same plot I had before. This is your excitation response, and the two photon excitation response would have some shape like this. Your linear response would have this airy disk, and we can do simulations and show that, yes, you actually get improved axial confinement, and you can get improved lateral confinement when you use those nonlinear microscopy techniques. Okay. So that's the fundamental background. So what we'll do now is, okay, what are people doing to engineer these devices? So we're going to talk a little bit about the basics of a multiphoton microscope, the basics of a nanoparticle plasmonic multiphoton microscope, and then looking at some quantitative imaging with nanoparticles for multiphoton microscopy. Great. Okay, so the microscope, in theory, this is from, there's a good review paper in 2003 from Nature Biotechnology. So if you're interested in this is a good place to start, and from there you can probably find a lot of good references and go forward, find some good forward references. So what you have here is basically a mode-locked laser, because remember, our signal goes with intensity squared. So if you want very intense light, and to get intense light, one way you can do that is to get very short pulses in time, high peak intensity, get higher signal back. So we have this pulse laser here, and we send it through some type of scanning system. So light will go through, and then we can raster scan our laser over our tissue. And it goes through our objective into whatever we're looking at. Then the light is going to be emitted back. This nonlinear signal comes back. We're going to have a series of filters so we can detect each of the different wavelengths of this nonlinear light coming back. And then from this, we can use a computer and kind of bring it all together and get this somewhat hyperspectral mapping of nonlinear emission as a function of position on the order of submicron resolution inside of tissue. Yeah.
Is the laser, what is the spectrum of the laser?
Oh, so it's like 10 nanometers, or 10 to 20 nanometers width at about 800 nanometer standard wavelength. Yeah. So yes, about 100 femtoseconds is the pulse width for these, and yeah, it's kind of Let's leave it at that. It's interesting, if you get shorter and smaller, you actually start creating thermal shock waves inside the tissue, and it kind of looks like the Death Star when you see an artery rupture and explode. So there's some issues about this thermal shock. So about 100 femtoseconds is nice. Okay, and so there's commercial. These are things you can buy. Olympus sells them, Nikon sells them, and here's an experimental setup. You have a Ti-Sapphire laser kind of sitting in the back over here, computer controlling it, and then it's basically just like a commercial confocal setup that they just modified and sell it for $500,000, 5 times the cost of the laser, just because they can. But they're selling a bunch of these, and hospitals are getting these now, pathologists are getting these now. Mostly those universities, researchers are working on this. And what you're looking at, okay, so there's some intrinsic stuff. Your cells autofluoresce. You have NADH inside of your cells, which is very common. All cytoplasm has that. So your cytoplasm is just going to glow. Your collagen, which is what keeps us together and stops us from becoming a pile of mush on the ground, that's going to become second harmonic. That's going to be very bright signals that are going to be exactly double the wavelength of your excitation source. And other things like hemoglobin, myelin, those 3-photon third harmonic scattering systems that you can use to identify these specific traits. And also we have intrinsic dyes. We can stick in fluorescein or rhodamine. And fluorescein is FDA approved. We can do fluorescent proteins, genetically engineered mice that have, you can specifically label certain neurons with a certain fluorescent protein. You can identify them in the brain. These are all techniques that we can use. And here's some examples. This is blood flow inside of a brain. You can see these little specks here correspond to the blood cells. If you park a line, like we're just parked right here, and you measure and you look at the function of time, you see the blood flow is going past. Well, if this is time in this axis, and this is x in this axis, we can find the slope here will give us velocity. And so therefore, we're actually measuring velocity of blood flow. People do this as far as a millimeter deep, or even up to 2 millimeters deep in the brain. You can measure blood flow in capillaries with some pretty high accuracy. We can basically do optical sectioning. This is an intact ovary, and basically we're looking at a slice through here without actually cutting it open. It's hard to see here, but there's a different spectral response on the outside, which is mostly like a collagen, elastin, connective tissue type of capsule around the outside. And then you have the structure of the ovary on the inside. You can see all the cytoplasm of them. And this is a rat tail tendon. You can see nice second harmonic generation signals from it. And so one of the projects we work on is sickle cell disease. It's a disease that's not actually very well understood or very well known. If you get a mouse that has humanized hemoglobin, sickle cell hemoglobin, there's lots of different morphological changes, but one of the most obvious one is the spleen. It gets really, really huge. This is 5 centimeters compared to a normal one's a centimeter. So basically you get a spleen the size of your pinky that's supposed to be the size of your pinky nail. And so we're looking at some, trying to find studies on this. We look at a lot of different tissue, brain, in the sickle cell mice. We have a bunch of going on, but I just want to use this as an illustration of stuff you see. So what you would see then in a healthy tissue, this green kind of corresponds to the cytoplasm of this tissue go through, and you can actually get some good. This is kind of poor resolution, but you see good structure of the cells, and you can learn about the organization and progression of this disease state. But if you look at the sickle cell disease, at the top, yeah, it looks pretty much the same, but as you go deeper, you see these little different nodules everywhere. And then we can go and look at each nodule individually and try to figure out what the spectral information is from each of those nodules. So we have these, we have a red one here, this red circle and this green circle correspond to these two, and we can compare it to the cytoplasm. And we can start, figuring out what are these things that are present. We can put them through other types of, techniques. We can do the sectioning and all that other stuff. And starting to find out that some of these are actually iron, that as the spleen is breaking down these bad red blood cells, it basically is forming these little blocks, little blobs of some, compounds with, lots of iron in them, and they're just sticking around the spleen because they're insoluble pretty much in everything else. And so, we're working with, medical doctors and immunologists and people who really are interested in these blobs of iron, and basically say, hey, we actually can see them inside of a living animal, and this is kind of one of the things that you can do with it. So what I just showed was we cut out the tissue, but you also could do this in living animals. So this is a mouse that's under anesthesia. We actually, gave him a little hat. So you remove part of the skull and put a glass slide over it, and you can shoot a laser into it and be able to see in 3D. And you can reconstruct, the vasculature of the brain. It looks something like this. And this is a preview for Professor Xu next week. He'll be talking a lot about being able to image deep through the brain and doing functional imaging. There's actually a kind of a cool paper where they put a mouse, in this microscope on a trackball. Has anyone heard of this experiment? There's plenty of great functional experiments going on in this, but this is a clever one, to tell students to explain like some things people can do. So you put this mouse on a trackball, and then you have his head immobilized and the laser is scanning to the brain, and basically you put a virtual reality display in front of it. In this case, they put the game Doom, the map of Doom, and then the mouse would run on the trackball, and his character would run through the maze, and then I guess he gets a reward if he gets to the end of the maze. And they were looking at basically, the neurons firing as the mouse is trying to figure out this maze, which is entirely virtual as he runs on this trackball. And I mean, it's just a clever experiment. It's just one of example of many that are going on that are kind of similar to this. And just a demonstration of what you can do real functional imaging. So this is like a living brain. Let's skip ahead a little bit. And yeah, you can actually see the blood flow. Astrocytes are genetically are RFP. There's some dye in the plasma that's not in the red blood cells. And you can zoom in, you can actually see the blood cells going past. And here again, the slope will tell you what the velocity of the blood flow is in this case here. And this is just one section of a living brain, so it's kind of a cool thing. Yeah. Oh, what's the field of view? Or what's
imaging speed or like this?
Oh, sure. So this was real time. So what you saw here was good. So depending on a pretty simple setup without using any type of resonant scanning mirrors, you can get a few frames per second, 5 frames per second. But if you use some special types of galvo mirrors, you can go to 30 frames per second. It then becomes a trade-off between, well, how much signal-to-noise ratio do you want over how fast you go? And if there's time, I'll talk a little bit about that trade-off, about ways you can try to get more photons out, increase your signal-to-noise ratio, and go faster. So the more photons you get out, the slower you go, the better your image is going to look. And so this is a part of the trade-off. You can also reduce the field of view and whatever. But yeah, so that was real time, and that was a couple frames per second. Okay, so if you're looking at an experimental microscope, nice clean Nikon. This is really what our labs look like. You got a PhD student sitting back there. You got the Ti sapphire laser and maybe an OPO if you're going for long wavelength. You got the microscope over here and the computer controlling it. And really, we're trying to figure out ways to make this more accessible, especially in like medical offices, doctors. What can you do with this? And one thing you can do is you make endoscopes, and so you can basically just put a fiber on a piezo actuator, so that you can raster scan this, and you have a fiber that's a dual clad fiber, so you have light can travel down the center core, and then when it hits your sample, it comes back out, and you have this outer cladding, which then will couple the light back towards your detector. So these are techniques people can do. You can package this into a little setup here. A couple groups are doing this. So I have one citation here and another group here, and later on I'll show you a third group. You can look them up if you're interested in this. And yeah, this is what it looks like. So what you do is you excite one axis at the resonant frequency of this cantilevered fiber, and so you have a very large and fast signal here. And then your other axis, you can just slowly scan, and you can basically eliminate a raster scan through your sample that way. And you can image things, and this is just from the paper where we showed. You can see this is collagen. This is basically lung alveoli. So you see little open spaces, and this is kidney, I believe. Oh no, this is colon, goblet cells in the colon. And then you also can make these rigid. So this is an example of, this is you can have a 20 centimeter long rigid lens, and you can use it to stick into soft tissue. And this is a kidney, and you can actually get the signal where you get, you see some of the capsule here in the second harmonic channel, and you get some high resolution images of the different cells, and you can do pathology, histology and pathology on it. Okay, so this is one of the things we want to think about is, okay, what can we do with plasmonics and this type of nonlinear microscopy? Because if this is something, if we really want to get down to the nanoscale, we can use nonlinear microscopy to get this depth information. And can we use plasmonics to get something that would be tens of nanometers types of chemical and structural information? Okay, so let's combine everything we talked about so far. We know gold luminescence. This is just something on its own. We can basically, with UV light, you excite electrons that eventually will recombine with the hole they just left from before. This has been seen for long, since the 80s, pretty much. Okay. We know that you can control luminescence by changing the structure and size of these particles. These are just quantum dots. So you can engineer quantum dots and get absorption in luminescence, which is greater than 1,000,000 times brighter than bulk, because you can have this confinement. You have really high quantum yields. So this is it's something that we can engineer. You can basically design a quantum dot that can emit anything from UV through IR. They have this is a good paper. It's kind of showing a design of how this can look. But now we have this really cool coincidence. Usually in nature, things don't work out cleanly. It's always like you have a trade-off, like, oh, I found this great technology, but it will kill the animal if I actually ever use it. Or there's this weird trade-offs you always come up with. But here's a really funny, like an interesting coincidence. We know that we can excite these gold particles about 400 nanometers, and they will you can get emission from them. We know these nanoparticles are small enough that you can have this plasmonic resonance in the near infrared. Okay, and so then if we have a lot of laser pulse, femtosecond laser pulses that are available between 700 and 800 nanometers that are already in bioengineering imaging labs, so we have these all around. So basically, we were thinking, okay, well, that means we can excite this, surface plasma resonance on these nanoparticles, which will have a very enhanced field of the surface, which will give us a very high I-squared for this nonlinear imaging, and have really bright 2-photon excited fluorescence, from these nanoparticles, much brighter than you could get, just on their own. And now we also have axial confinement because this is a nonlinear process. And so there's been actually it was first shown. I have the paper here. It was first shown in '86. They basically just showed that it happened as part of it works. There's no scale. There's no quantitative data. They just acknowledge the fact that they can see this. But lately, people have been revisiting this and really trying to measure this and understand how this works. So this is a paper where they made these these nanorods in the order of 50 nanometers. They show if you plot power versus output power, you get a log plot slope of 2. So it's a 2-photon process. If you plot the 1-photon absorption, this is the surface plasma resonance, and you look at the 2-photon emission, you get a peak of 2-photon emission when you're exciting at the surface plasma resonance. So the surface plasma is getting excited, and when it's excited, it's doing a 2-photon absorption process. And they do some cool stuff, and they look at, well, what's the polarization effect? These are rods. And if I align my electric field in this axis, I should get some resonance. In this axis, I shouldn't. And they pretty much show that. They're basically on different particles with different polarizations. You can actually show a very strong response based on polarizations. So we actually are saying, okay, we're exciting a surface plasmon resonance in nanoparticles at the surface plasma resonant frequency that is, has polarization effects and a 2-photon response. And this response we're getting back, this is brighter than rhodamine, which is a very commercially available, very bright dye. And so what people have done, this is Xingdi Li's group at Johns Hopkins. They make this very similar type of endoscope that we saw earlier, and they want to look at these nanoprobes. So they create these nano cages that are also in the order of hundreds of nanometers. The surface plasma resonance is 790 nanometer, right around this high sapphire laser, and you can look at them and you see them in your endoscope. They then do the step where they functionalize the surface, and they put these different antibodies on here that will attach to tumor cells. And then they image the tumor cells on their own. There's nothing there. You put in these antibody-labeled nanoprobes, and they light up your tumor cells. And each of these probes we know are localized at about 100 nanometers. And on top of that, we have this very enhanced field on the surface, so you can try to do some other stuff. Maybe you could put some fluorescent proteins or something on the surface that when it binds is quenched, or you could do some of the surface chemistry here that could be really interesting for people who study membranes, for example. Okay. So far, we're just saying we got good pictures. We got physiological information. We have structural information. For the next, I guess, 10 minutes or so, I'll talk about, well, how can we get chemical information out? I want to know specifically at each point, what is the partial pressure of oxygen on the order of 1 to 3% accuracy, 500 microns deep in brain in a living mouse. Okay, this is my goal is, which right now is not really too obtainable. So the strategy we're using is fluorescence lifetime microscopy. And the idea here is that if you have a fluorophore that can be quenched by some localized molecule, the amount of light that comes out of this fluorophore and the rate of which this light comes out is going to be a function of how much of this quencher is present. So let's say this fluorophore is quenched by oxygen. I put a lot of oxygen present. I will reduce the emission. And the reason why this happens, basically what's happening is you're exciting a triplet oxygen to singlet oxygen, and this decay path is very, very, it steals this energy through its collisional process. And so the phosphorescence emitted by your fluorophore is going to decrease as the electrons that normally would be in this excited state are now going to be stolen pretty much by the oxygen, and you won't see this. So the two things that will happen is the intensity will go down, and the lifetime of electrons that are present in this excited state also will go down. It will be much faster. So if I measure the lifetime in the standard without oxygen present, you might have some oxygen-deprived lifetime. You put oxygen present, you see this decay change, and we can measure this, and we can quantitatively correlate this decay change to oxygen that's concentration. And this is just a demonstration of what it looks like. So you can have some dye, and so you put nitrogen in, and it's going to glow. This is just a dye with the UV light on it. And if you put oxygen in, oxygen will quench the dye, and this response will go away. This is just what it looks like in real life. Okay, so recently there's been some really great paper. So this is basically David Boas at Harvard did the imaging side of it, and Sergei Vinogradov over at University of Pennsylvania, he did the probe development. So they injected these probes into the mouse. They did 2-photon microscopy to get the same structures we were looking at earlier of the vasculature of the mouse. And then they said, okay, let's measure the oxygen concentration. So they move the laser to these specific points. They park it there, and they do these measurements, these lifetime measurements. And then from there, they can measure, okay, I know what the oxygen levels are at each of these points. And then they can do a 3-dimensional mapping reconstruction of what it looks like. So this is a really powerful technique, and you can park it there and do some functional imaging. But in reality, you're limited by the amount of time it takes to take this. This takes several minutes to capture this data. Each of these points can take up to 20 seconds. So if you want to do like a full frame imaging and get information throughout all the vasculature, you have to find another way of doing it. So you're fundamentally limited by the speed at which the photons come back, by the rate at which you can look at them. And in order to get to faster frame rates, to be able to image this faster, you need to be able to spend more time on each pixel, or you just need to crank up your power. But at some point, you crank up your incident power so high, you saturate all your fluorophores, and no matter how much higher you pump it, you don't get any light. You don't get any more light back, so you're not improving its noise ratio. Okay? So that's like the fundamental problem. And so this is just a slide I put through in here to kind of show like how do you measure lifetime. Well, one way is you just put in a pulse, and you can just measure. You can actually just measure the decay using time-correlated single photon counting. Another way you could do this is if you think about your fluorophore as basically an RLC circuit, or any type of circuit with a time constant, you know that the frequency response of the circuit is going to be, as you approach this decay time of your circuit, you're going to have a decrease in amplitude of your signal through this filter, and you're going to have a phase offset. And so it's the same math that goes into this. You know, you can work this out, and you can actually measure. If you measure this phase offset, you can back calculate out what was the lifetime of your circuit or of your fluorophore. This is the technique, you know, that we'll mostly use. And so what you could do then is just mostly showing that if you show this if you measure this offset and phase, you can back calculate out the lifetime. Okay, so what we do then is we think about this like an AM radio. Let's excite all the points along here at a different RF, a different modulation frequency on the order of kilohertz to megahertz. And then we can scan this illuminated line across our sample. We collect all the light back. We demodulate at all these frequencies, and we can calculate both the magnitude and the phase. And then from the magnitude, we can get to the intensity of the point. And then from the phase, you can get to the lifetime. And you can do the full frame. And so this is images showing the full frame. So we can integrate for longer, and we can we can measure the lifetime with more accuracy. We can do different steps throughout the brain for these images. And you know, if we can, they also show that they're actually in there. So this is the vessel without anything, without any dye in it. This is our intensity map from our modulation system. And here's the lifetime map. And you can see, again, you can see the bright cell walls as very low lifetime because there's just fluorescent proteins and collagen and elastin. And then inside the vessel, you have the longer lifetimes corresponding to our dye that's currently flowing through this mouse that's alive's brain. Of course, we're still limited by our pixel, so we need to work on new dyes. Now I guess we'll just wrap up showing, you know, some stuff we're working on is trying to make these nanoprobes or nanoemulsions so we can measure dyes. There's some common oxygen-sensing dyes out there that are not water soluble. So if you can surround them with some surfactant, which is what we're doing here, we have our dye, which is not water soluble, surrounded by surfactant, so it becomes water soluble. You have this hydrophilic fingers sticking out, so it can kind of float around in your water. So one way to do it, another way we can make these about 200 to 400 nanometer diameter silica cages. We can load dye in the middle of them, and then oxygen is permeable through the silicate cage, and we can use those as dyes. We can measure the 2-photon emission cross-section. So this is how much light we get out as a function of excitation wavelength. And we show that, you know, these types of encapsulations are as bright as rhodamine, if not a little bit brighter, much brighter than fluorescein. So this is a useful dye for 2-photon measurements. We can cycle a cuvette with oxygen. So we cycle oxygen, which is measured with this dashed line here with a commercial probe. We can continuously measure our lifetime and then make a correlation plot so we get a nice calibration. So we know for measured lifetime what's the partial pressure of oxygen. We get about 1 to 3% of the full scale accuracy. And if you show what the sensitivity is of this probe, it's, you know, as good, if not better, than all the other probes, which are ruthenium based and oxygen and water soluble. So we now have this very sensitive, bright 2-photon probe. We can use it and look inside. This is, again, a three-dimensional view. So we're going through a living mouse in 3D from the surface of a blood vessel to the top of the blood vessel. So you kind of see this, like this shape kind of grow, you know, expand and shrink, which is the blood vessel growing. Here is a mouse macrophage loaded up with these pebbles, so these silicate cages that have dyes in them. Each one of them is 100 nanometers in diameter. This cell is interesting because it's actually going through splitting. So you have 2 nuclei present. And so we can look at each of these probes individually. And here's an experiment where we tried, the resolution is not so great here, but each of these are our probes. And we have the red ones are the oxygen sensing probes. The green ones are hydrogen sulfide sensing probes. We put in hydrogen sulfide into the cell culture medium, and you actually can see the decrease in intensity of the yellow, which is the hydrogen sulfide, and oxygen stays as bright as it was before. Just kind of showing the selectivity of these cells. And yeah, so I think that kind of wraps up my talk, trying to give an introduction to both plasmonics and nonlinear microscopy of ways of getting chemical information on the hundreds of nanometers scale in living tissue. So kind of we talked about, you know, what we needed, the physics that we had to learn and understand, the knobs we possibly turn. And then this is a good summary of what we talked about. We talked about just what the big microscopes look like. We cut out tissue. The bigger microscopes look like when you have a, you know, in brain imaging. We talked about endoscopes and plasmonic endoscopes. And finally, we talked about this quantitative chemical imaging using encapsulated dyes and nanoprobes. Okay, and yeah, we have. So I'm new at NerdAIM. I just started years ago. So I have 3 grad students who 2 of which did work on this type of project. And I have my collaborators at Cornell, which some of this work was, for my postdoc. And we also work with, we have to work with medical schools, University of Connecticut, Indiana University, Ken and Roger. I work with a lot of people at NerdAIM, but for this work here, I work with a chemical engineering professor who does some encapsulation work. And the funding that keeps the lab alive comes from Walter Cancer Foundation, some internal money from NerdAIM, USDA, and Homeland Security. So great. So I think that's it. And welcome any questions or discussion about the technology. Yeah. Yes. It's 2 photons. Yes. Yeah, that's like a millimeter of X. What is the probability of coming back those photons in the same? Sure, okay. So this is a great question. So she's asking, that basically we're imaging 2 photons. We're putting these photons in, they're exciting something, and then they're coming back. And what's the probability that photons will actually come back through the same path in order to be collected by our system? Scattering is a big problem. So this is one of the cool hidden things about multiphoton is that first off, for phase matching, only statistically almost it's very unlikely that any photon that scatters will cause a 2-photon event. So the 2-photon excitation point are only ballistic photons. So only the photons that did not scatter to get to the point are the ones that emit a signal. And then you say, okay, well now it emits, it's just going to glow and bounce all over the place. Since I am basically multiplexing this in time, I know that every photon I collect, no matter where it scatters to or from, had to have come from this localized point that was created only by ballistic photons. So we just collect all the light back. We don't even descan it. We have a big single area detector. So no matter what path the photon gets to get back to my detector, we knew it came from this point that was created by ballistic photons. So it's basically like confocal microscopy for free, that you don't need to pinhole because of the ballistic photons created this case where only excitation comes at a localized volume. And so it's a great question. So thank you. Yeah, so that's the thing. People who work with confocal microscopy, autofluorescence is annoying, but we like autofluorescence. We actually because it's a way of us to do label-free spectroscopy. But so yes, there is a problem with autofluorescence, but people are trying to leverage that and trying to use the autofluorescence as a contrast mechanism. But yeah, autofluorescence can be pretty bright. Yeah, good questions. Thank you. Oh, so you mean dispersion and all that stuff? Yeah. So this is this was one of the big problems with the endoscope. She's asking, how do we compensate for the difference in wavelengths? We have all these optics in there that are basically sitting in infrared light and then focused down to a point, and then we have visible light coming back. So the original idea was, and actually we tried this, it took us a while to get working. I had a picture here. So this is the problem here is, okay, the light, if you're trying to focus down to this point, this focal length of these lenses for 1 micron can be very different than it is for 400 nanometers. What actually was happening is it's like a bifocal lens. So we actually use the center part for the infrared light and the outer part for the visible light. And they're coated with anti-reflection coatings, different coatings, in order for it to compensate back. That's what we ended up doing was proposed, and we eventually got there. In the end, we just brute forced it. This dual clad fiber has 100 microns, actually a couple hundred micron outer cladding. So whatever problems we got from this lack of focus, we just didn't collect very efficiently, but we collected enough that we still could see. And so that was kind of the way. But yeah, I mean, that was actually, when this proposal was written, the innovation was, how do you do this? And so we were trying to, basically in the clean room, with photolithography, you define, this, because these are all things, a couple millimeters in diameter. So you have to, do lithography on a curved surface, and you got to sputter on your different coatings, and it's, it's difficult. But that's a great question.
So So in your oxygen quenched probe, if you're exciting oxygen to its singlet state, are you getting any sort of PDT effect?
Oh, yeah, so, well, oxygen is a singlet state has lots of problems with that. Yeah, so that's actually one of the, you know, reactive oxygen species we were asking about are very damaging to your cellular stuff. And additionally, they also fluoresce at 1250-ish nanometers. So luckily for us, 1250-ish nanometers are past the cutoff of our detectors, so we won't see that fluorescence. But that's an area I'm actually interested in. You know, I want to start working a little bit on can we use this for therapy? Actually, you know, there's ways of trying to increase oxidative stress in tumors to the point where you can trigger cell death in the tumor, but you don't trigger cell death for the healthy cells. And so this is the way that, you know, Naomi Hollis has a whole company that does gold nanoparticle cancer therapy. That's what she does, you know, and they use gold. They use gold nanoparticles like this, and they excite this singlet oxygen, and this reactive species adds oxidative stress to the tumor cells, and they die, and the healthy cells can live. Because you don't want to just blow up a tumor cell, because it will just end up all over the place. You can cause metastasis, and it'd be bad. So you want it to die itself, and. But yeah, so I mean, this is kind of newish stuff, especially this oxygen quenched imaging. We're using mouse models, and so you know, we're not too worried. These mouses have so many other diseases that we're not too worried about what diseases are, you know, what's going to happen to the oxidative stress. But eventually, you know, that's something we should quantify and look at. I believe that's what you're looking or asking for, or is that? Yeah.
And it would be interesting too, because a lot of people try to do vascular PDT anyway to try to shut down, you know, the nutrients to the tumor. So you're sort of already doing that in a sense.
Yeah. So I think actually we have one of the newer proposals coming out is looking at breast cancer metastasis to the brain, and basically when you have these breast cancer cells start lodging in the brain, only less than 1% of them actually stick. And we want to study, well, why do that 1% stick and other ones don't? And then once it sticks, what metabolism is going on? And once we understand that, can we just stop them from sticking? It's kind of a way of preventing breast cancer metastasis. You know, it's just fundamental biology, and then there's a lot of science behind that. And then also, you know, this oxidative stress. Can we measure this ratio? You know, that's what I do. Ratio of oxidative species, reactive species to, you know, the triplet species, and kind of get some information, you know, quantitative at that level. This is all photophysics. Yeah, great. That's a great question. Thank you.
All right. Well, let's take a break.
I think this is the first time you've seen my my new stuff.